Hello, namaste, and welcome to Simply Biology with Manisha. In this video, I am going to explain the process of isolation and purification of DNA from living cells. First of all, I am going to describe how many types of DNA are commonly isolated. In general, three types of DNA are routinely prepared in laboratories, namely total cell DNA, extrachromosomal DNA, and phage DNA. I will explain extrachromosomal and phage DNA isolation in my upcoming videos. Here I am going to describe how total cell DNA isolation is performed. Overall, the process can be categorized into four stages. In the first step, a cell culture or tissue sample from the desired organism is obtained. In the second step, cell lysis is performed. After this, DNA from the lysed cells is purified and concentrated. In cell lysis, I will gloss over the process of mechanical and chemical lysis. I have broadly classified the process of DNA purification into different steps named organic extraction, enzyme digestion, ion exchange chromatography, and use of silica and guanidium thiocyanate. Usually, DNA is concentrated by precipitation with ethanol. All the procedures that I am going to delineate in these videos are very basic and very commonly conducted in laboratories. So please keep in mind that there are other methods as well for DNA extraction that I have not included here for the sake of simplicity. In this video, that is the part 1 of this series, I will elucidate this process till the step of cell lysis. I will be discussing DNA purification and concentration in part 2 and part 3 of this series. Let's begin the topic by looking at the types of DNA that are commonly extracted by researchers. The first type is total cell DNA, which can be isolated from bacteria, plants, animals, or any other desired species. The entire DNA present in a cell, for example, genomic DNA or nuclear DNA, and extranuclear DNA, like the DNA contained in plastids and mitochondria, all of this constitutes the total cell DNA. The second type is extrachromosomal DNA, which includes plasmid DNA in bacteria, mitochondrial DNA in higher organisms, and plastid DNA in plant cells. For the major part, the method of plasmid DNA extraction is similar to the procedure for total cell DNA isolation, the only difference being that at some point we have to ensure that the plasmid DNA gets separated from the chromosomal DNA. The third type of DNA that is routinely isolated is phage DNA. Here also slight modifications are made in the procedure in order to remove the capsid of the bacteriophage. In this video, I am going to describe total cell DNA isolation. I will be discussing the other two types of DNA extraction in my upcoming videos. I am taking a typical bacterial cell as an example for the illustration of total cell DNA isolation. In the first step, the desired bacteria are inoculated in a suitable liquid culture medium. The bacterial culture is allowed to grow in optimal temperature and pH conditions until the desired cell density has been obtained. Then, the liquid culture medium is centrifuged, following which all the bacterial cells come down in the tube, forming the pellet. Subsequently, the cells are lysed so that all the cellular contents are released outside, giving us the cell extract. In the purification step, all the contaminants are removed, leaving only the DNA in the cell extract. In the last step, this DNA solution is concentrated. First, let us see how the cells are harvested. The bacterial culture is centrifuged at a moderately low speed so that the cells come down to the bottom of the tube, forming a pellet. In the next step, These cells are lysed to obtain the cell extract. Let's start with understanding what cell lysis is. Typically, the bacterial cell envelope comprises two layers, the plasma membrane and the cell wall. Often, the cell wall is made of peptidoglycan. In some bacteria like Escherichia coli, an additional outer membrane is present outside the peptidoglycan layer. This means that the bacterial cell wall may be composed of only peptidoglycan or of peptidoglycan as well as outer membrane. The process where both layers of the cell envelope, that is, plasma membrane and cell wall, are fragmented to release the cytoplasmic contents is known as cell lysis. Let's take a look at some techniques commonly used for cell lysis. The first one is the physical method where mechanical force is applied to break open the cell envelope. Nowadays, researchers do not use this technique frequently, but this method is definitely favored for plant cell lysis. The second method is chemical lysis, which is preferred for the purpose of DNA extraction. Some chemicals which are commonly used here are lysozyme, EDTA, and detergents. Please note that it is not necessary that only one of these methods be used at a time for cell lysis. Researchers may put together 2 or 3 chemicals, out of which one attacks the cell wall and the other the plasma membrane, or they may use a combination of both physical and chemical methods for cell lysis depending upon the organism that is being investigated. Following cell lysis, the broken fragments of cell wall and plasma membrane that are left in the cell extract are collectively known as cell debris. So at last, the cell debris is removed and only a clear solution of the remaining cellular components is taken forward for the next step. First, let's study how chemical lysis of the bacterial cell wall is done. Lysozyme is used for this purpose, an enzyme which breaks down peptidoglycan present in the cell wall so that the cell wall loses its rigidity. Lysozyme is naturally present in egg white and in bodily secretions like milk, saliva, and tears, where its original role is to break down the cell envelope of any bacteria that comes into contact with cells of the organism that is producing this enzyme. In the case of plants, where cell wall is mostly made of cellulose, lysozyme is not able to lyse the cells. That's why mechanical lysis is preferred here. First, we take the desired plant tissue, for example, leaf. In the presence of liquid nitrogen, we grind the leaf using mortar and pestle. Liquid nitrogen appears like boiling water with steam coming out of the liquid, but it has an extremely low temperature of around minus 196 degrees Celsius. When cells come in contact with liquid nitrogen, they freeze instantly and on grinding are easily pulverized. The second advantage is that at such a low temperature, DNA degrading enzymes called DNases are inactivated, which helps in extraction of intact DNA molecule. After grinding, the frozen powder is immediately mixed with DNA extraction buffer. Another chemical which is usually added to the lysis buffer is ethylene diamine tetraacetate, in short EDTA. It chelates divalent cations, in other words, positively charged ions like magnesium ions. Cell walls of many bacteria contain lipopolysaccharides, which remain in a stabilized state with the help of cations like magnesium and calcium. Upon formation of the cation EDTA complex, holes are left in the cell wall at all those places where the cation was originally present. This destabilizes the structure of the cell wall so that the cell bursts open and all the cellular components are spilt out. In these cellular components, the cell's DNA is present, but along with this, DNases are also present. The DNA degrading enzymes about which I told you earlier as well. Now, this is a problematic situation because our aim is to extract intact DNA molecules. The solution to this problem is that these DNases require cofactors in order to be functional. And generally, these cofactors are magnesium cations. But we know that these magnesium cations are already complex with EDTA. So, because the cofactor is not available, DNA's activity is hindered and DNA is not degraded. Thus, EDTA contributes in cell lysis by these two means. Ordinarily, lysozyme and EDTA are sufficient to lyse bacterial cells, but a detergent, for example, sodium dodecyl sulfate, commonly called as SDS, is also added to the lysis buffer. We know that the cell membrane is composed of lipid bilayer. The detergent molecules replace these lipid molecules in the bilayer and form micellar structures with these lipids, due to which the cell membrane structure is totally disrupted. Now, in the case of animals, since the cell wall is not present, treatment with detergent alone is enough to bring about cell lysis. Thus, EDTA is not required for lysis of animal cells. However, please take note, EDTA is used here not for lysing animal cells but for inhibiting the activity of DNases. After lysis, we need to eliminate the insoluble cellular components like fragments of the disrupted cell wall. Hence, the lysed cell extract is centrifuged, following which the unwanted cell debris comes down in the tube, forming the pellet, and we get a clear solution in the supernatant which contains the lysed cell's DNA. But, too bad, this solution also contains protein and RNA from the lysed cells. So after cell lysis, we move on to the next step where protein and RNA are removed from the cell extract to obtain a pure solution of DNA. This I will explain in part 2 of this video series. Now, let me ask you one question related to the topic I've just discussed here. Can lysozyme be used for plant cell lysis? Type your answer yes or no below in the comment section. You can also write your feedback and queries in the comments, and please like and subscribe to my channel Simply Biology with Manisha and click the bell icon to get a new video update. Thanks for watching. Goodbye for now.